Hi, in this video we are going to study about glycolysis. Glycolysis, also known as EMP pathway or Embden-Meyerhof-Paranis pathway. It is a process in which 1 mole of glucose is partially oxidized into 2 moles of pyruvate in a series of enzyme catalyzed reaction. Glycolysis occurs in the cytosol of all cells. It is a unique pathway that occurs in both aerobic and anaerobic condition, and it does not involve the use of molecular oxygen. So, glycolysis is oxygen independent. The glycolysis completes in about 9 steps, in which the glucose molecule, which is a 6 carbon compound, gives 2 molecules of pyruvate compound. The process of glycolysis starts when glucose enters into the cytosol and it is immediately converted into glucose 6 phosphate. This is the first step of glycolysis. The reason behind the formation of glucose 6 phosphate is that the glucose cannot be escaped out of the cytosol. So, steps of glycolysis. Glycolysis involves about 9 steps to complete the whole process of glycolysis. First step is called as phosphorylation. Why? Because glucose is phosphorylated by ATP to form a glucose 6 phosphate. This irreversible reaction is catalyzed by enzyme hexokinase. Hexokinase is present in all the cells of all the organisms. Hexokinase requires divalent metal ion such as Mg2 plus or Mn2 plus for its activity. So here there is a utilization of ATP molecule. The next step. The next step is the isomerization. What is isomerization? A reversible rearrangement of chemical structure called isomerization moves the carbonyl oxygen from carbon 1 to carbon 2, forming a ketose from an aldose group. Thus, the isomerization of glucose 6 phosphate to fructose 6 phosphate is a conversion of an aldose to a ketose. This step is catalyzed by an enzyme phosphoglucoisomerase, or it is also known as glucofructoisomerase. Step 3, which is a phosphorylation step. It is also an investment stage. So here fructose 6 phosphate is phosphorylated by ATP to fructose 1,6 bisphosphate. The prefix bis in bisphosphate means that two separate monophosphate groups are present, whereas the prefix di means that two phosphate groups are present and are connected by an anhydride bond. This irreversible reaction is catalyzed by a allosteric enzyme phosphofructokinase, or it is also known as PFK1. This step, which is a step 3, it is also an investment phase where ATP molecule is utilized. One ATP molecule is utilized and one ADP molecule is formed. So the next step is the cleavage. The fructose 1,6 bisphosphate is cleaved to produce two 3 carbon molecule glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by the enzyme aldolase. So, here occurs the cleavage process. In simple words, we can say that one fructose 1,6 bisphosphate is cleaved or divided into two 3 carbon molecule glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by enzyme aldolase. Here occurs the isomerization. How? Only one of the two triose phosphate formed by the aldolase glyceraldehyde 3 phosphate can be directly degraded in the subsequent reaction steps of glycolysis. However, the other product, which is dihydroxyacetone phosphate, is rapidly and reversibly converted into glyceraldehyde 3 phosphate by the enzyme triose phosphate isomerase. Step 5. The two molecules of glyceraldehyde 3 phosphate are oxidized. Enzyme glyceraldehyde 3 phosphate dehydrogenase catalyzes the conversion of glyceraldehyde 3 phosphate into 1,3 bisphosphoglyceric acid or 1,3 BPG acid. The reaction occurs in two steps. That is, first, the oxidation of aldehyde to a carboxylic acid by NAD+, and the second is the phosphorylation of carboxylic acid by inorganic phosphate, not by the ATP. Next step. Step 6. In this, high energy phosphate group is transferred from 1,3 bisphosphoglyceric acid to ADP. This step is catalyzed by the enzyme phosphoglycerate kinase. The formation of ATP is referred to as substrate level phosphorylation. In case of substrate level phosphorylation, synthesis of ATP or GTP occurs directly by transfer of a phosphoryl group from high energy substrate to ADP or GDP. This is the first payoff step in the glycolysis, where one ATP molecule we get, or rather we say two molecules of ATP. Here we get step 7. Step 7 says that the remaining phosphate ester linkage in the 3-phosphoglycerate, which has a relatively low free energy of hydrolysis, is So, step 7. The remaining phosphate ester linkage in 3-phosphoglycerate, which has a relatively low free energy of hydrolysis and is moved from carbon 3 to carbon 2 to form a 2-phosphoglycerate. Simply, it means that the 3-phosphoglyceric acid converts into 2-phosphoglyceric acid, which means there is a hydrolysis process which is occurring in presence of enzyme phosphoglyceromutase. Very simple steps where the hydrolysis occurs. The second last step of glycolysis, which is the removal of water from 2-phosphoglyceric acid, which creates a high energy enol phosphate linkage catalyzed by the enzyme enolase. Here, 2P, 2-phosphoenol pyruvic acid is formed from 2-phosphoglyceric acid by the enzyme enolase. It is the second last step of glycolysis, and the last step. Finally, in the presence of enzyme pyruvic acid kinase, the 2-phosphoenol pyruvic acid converts into pyruvic acid. During the conversion of ADP will be phosphorylated into ATP. So here once again there is a phosphorylation, or there is a payoff stage. So the formation of pyruvic acid indicates the end of glycolysis, and this is how the glycolysis occurs in 9 steps. The last step is also the payoff stage where the ADP is phosphorylated into ATP. So here we get two molecules of ATP. So in total we get about 4 ATP